Hello, I'm Dr. Mileidy Giraldo, Global Lead for Genomics R&D at Lenovo HPC and AI. Today, I'm going to tell you how genomics optimized servers are accelerating COVID-19 discovery with high throughput analytics. As of today, Johns Hopkins University reports over 7 million confirmed cases of COVID-19, having led to over 400,000 deaths in 188 countries around the world. And with the statistics still rising, understanding the virology of the novel coronavirus and developing an effective vaccine or protective measure are front and center in the minds of biomedical researchers and clinicians worldwide. Today, I'm going to show you that HPC clusters that are optimized for high throughput analytics are crucial to the success of these efforts. But what type of insights are COVID-19 researchers interested in? Well, there are many unknowns regarding the virus, the mechanisms of infections, or the immune response it elicits in the host. But what we know is that researchers are tackling this unknown from many different angles. Here's a list of some of the highest priority efforts in the biomedical and pharmaceutical space. We see research being done in vaccine design and improving diagnostic kits, identifying determinants of susceptibility, looking at virulence, tracking the spread of the virus, the origin of the virus, identifying drug targets, and designing protective measures. What all of these seemingly different research approaches have in common is that genomics analytics is one of the first steps used to derive scientific insight. So, for example, researchers might use genomics analytics to focus on identifying genetic differences. Let me give you an example: severity of infection. See, one of the biggest mysteries in this disease is that in some people it presents as a mild cough or it's even asymptomatic, whereas in some people it turns into pneumonia or even a fatal case. So to understand why that is, researchers might perform sequencing and genomics analytics on the viruses of the genomes of the mild case and compare the genomes of those of the pneumonia cases. And for illustration purposes, I'm showing you a sample of, I'm color coding this of, green, a lot of green viruses here and red viruses. But this is something that cannot be so easily done, and it's a difference that can be distinguished visually. In fact, the differences are genome deep. And what the researchers have to do is look at the viruses of the people infected with a mild case versus those of pneumonia. And see, the reason we want to do this, and if in fact we were able to find this, that the dominant variant in the mild cases differs from the pneumonia case, it would be a breakthrough because clinicians and hospitals could use the information to triage patients. So if we could quickly figure out that a patient coming in has a pneumonia-prone strain rather than a mild case, we would be able to prioritize care and even treat the infection differently for the different cases, much like we do with the flu. Right? So that's one type of research. But other researchers, instead of looking at the genomes of the viruses, might look at the genomes of the patients instead of the two groups. And so what they might find is that the mild cases share in common some factors that we call innate protective factors. And what that means is factors that confer protection somehow. And that's an insight that is of extremely high interest because if we find those protective factors, we can retro-engineer the factors and confer that same protection to people who don't have it. So it would inform potential treatments. Okay, so I've given you examples here of how genomics analytics is used to find genetic differences, but there are other types of research that instead focus on the similarity, and vaccine design is one of those examples. So this is something we've all heard about before, where the viruses mutate over time. But what does that mean? It means that initially you may be infected by a single virus, a single copy of the virus, or maybe a few, and the population in the beginning is very homogeneous. But over time, as the viruses replicate, they tend to change. And so what we do in vaccine design is compare as many of the viruses as we can. And in this diagram, I'm just representing a virus and next to it, its genome. We would align them and look for regions that don't change, that regardless remain the same in all viruses. And we call them conserved regions. The reason we want to find those regions, and those regions in fact become potential vaccine targets, is because then we can train the immune system to recognize those regions that don't change and to mount an immune response next time it sees them. And that's how we keep you healthy. And these are the basics of vaccine work. Now, you might appreciate then that the bigger the sample here, the more viruses we can look at, the better our chances of finding an effective vaccine. And see, many of the vaccine trials and vaccine efforts fail because oftentimes the sample that is looked at, and say, for example, the regions we thought were conserved were not really once we give them out to the entire population. So if you think of a target, if that target keeps changing, then that effort will fail. And so again, very crucial for vaccine design to be able to enable high throughput work on a large scale sort of analysis. All right, so far I've given you only two examples of the sort of research that's being done in this space, but keep in mind that there are other types of research that are also being done leveraging genomics analytics. But what is the role of HPC here? How can HPC enable the discoveries in COVID-19? So it turns out that in life science in general, and very specifically for COVID-19, it requires HPC that has the following characteristics. On the one hand, HPC that supports high throughput volumes. So as I've shown you in the two examples before, COVID-19 work requires research at the population level. And what that means is no longer are researchers doing the sort of research we did 10, 20 years ago, which we only looked at a few genes and a few people where we could do things manually. Now we're looking at many genes, an entire genome, on many virus particles, on many infected people. So you can see the scale here is much larger. And what that means is that in practical terms, they'll be analyzing more genomes and needing to do it more quickly. And as I mentioned in the previous slide, the bigger the samples, the better our chances of finding an effective vaccine or treatment. So for those of you supporting the HPC effort for the scientific computing teams, a call to action here would be finding ways to increase the throughput capacity of the workflows your researchers are running. Another one is prepare to have to increase the storage capacity because this sort of work requires a lot, it's producing a lot of data, and it will require fast storage capacity at that. Another factor that is important is having HPC that accelerates secondary analyses, what we call the omics analytics. And let me explain what I mean by that. I'm going to show you a diagram that shows the entire discovery process, all the way from that sample from saliva, from spit, all the way to what we hope to get to, which is the COVID-19 discovery. This is done, in fact, in three phases. The primary analysis, and when you hear your researchers talking about this, what it means, primary analysis is taking that biological sample, putting it into a sequencer, and getting a computable form of that data. Now comes the HPC portion of it. The secondary analysis, this is where your genomics, all the omics, genomics, transcriptomics are performed to do the sort of analysis that I showed you in the previous slides, the two examples. That happens at this phase. Now, the output of that, no, it doesn't stop there. The output of the secondary analysis becomes part of the tertiary analysis. And so we take it further, and then researchers do biostats, machine learning, AI, molecular modeling, and so on, to then arrive at that application that becomes a discovery. Now, by far, when looking at this entire process, one of the largest steps really slowing down the progress here is the secondary analyses. So a call to action for the HPC support teams is to set up robust HPC infrastructures that optimize these secondary analyses. And I'm going to come back to that in another couple of slides. But before, let me mention two other factors that are important as well. On the one hand, increasing usability. So see, to set up an NGS, a next generation sequencing operation, when you do this, you face a steep learning curve. And the reason is that this is not traditional HPC. In traditional HPC, we have a code that we farm out to many nodes. So we generally tend to spend time optimizing the infrastructure on which that code will be run. In genomics, you have workflows like the GATK workflow by the Broad Institute and some others, but it's still the same. You have over 30 different tools all running in a single node. They all have very different profiling characteristics, and they work sequentially. Some of them are single-threaded, some of them are multi-threaded, some of them are memory hungry, others are not. So it's a very tough HPC problem to optimize an infrastructure to benefit the entire workflow, to benefit all the different steps. So much that it requires specialized HPC and subject matter expertise working together to really make any headway. And in fact, because of this, it's something that is seldom or never undertaken at data centers. So the call to action is, any, high performance computing teams supporting researchers that can do anything to lower the usability barriers in terms of the pipeline setup and optimizations of the hardware will make huge strides in helping the researchers greatly decrease time to scientific insights, because this is the meat of that bottleneck I was mentioning. All right, another thing to keep in mind is that anytime you work with human data, there are, you know, data privacy concerns. So providing a platform that ensures appropriate user authentication and controlling data access is a must. All right, so let me revisit this bottleneck because it's one of the biggest challenges in HPC and a place where we can contribute. So when we go out and, in talking to people doing this for a living, what we see is that people running this sort of workflow on-prem is taking them 150 hours. That's about a week to process a single human genome. In about 4 hours to process an exome. An exome is a smaller data set of the genome. And so given the scale and given the urgency of this sort of research, this is a scale that has to be addressed. Cloud computing solutions can provide the same analysis in about 30 to 40 hours. Now, back in 2017, Intel addressed this problem and developed an architecture to reduce this greatly, down to 11 hours for a whole genome. Then last year, following the steps of that work by Intel, we created a solution and we found an architecture that we called GOAST. It stands for Genomics Optimization and Scalability Tool. And this architecture no, includes no changes to the code, but it's hardware and system base, and it reduces um, the analytics. It reduces that execution time down to 5.5 hours for a genome or 6 minutes for an exome. That's a 27 to 40x speed up. Now, because of this, we were awarded the first Intel Select Solution certificate for genomics analytics by Intel. And it was a great accomplishment because we're reducing this, but then we think we can do better. And in fact, after the onset of the COVID-19 pandemic, we cranked up our in-house optimizations. Our Lenovo Genomics R&D team took this task because this is a place where we think we can contribute in the battle against COVID-19. And so again, we had in mind that we had to search for the best hardware and system recipe we could to support these four characteristics. High throughput, we had to deliver accelerated speeds, increase usability, and an environment secure and data privacy. See, our goal for this work has been to focus on the technology so that our collaborators and customers don't have to, and they can instead focus on the science. But to do that, it required performing a large-scale systematic permutation study. So we looked at and tried to figure out what are the factors that can affect performance of these workflows? What is it that is slowing down? And so we looked at hardware, software, workflow setup strategies, and many, many system tuning parameters. We looked in terms of hardware, we look at processors, many different options, storage options that you see listed there, memory from 300 gigs to all the way up to 12 terabytes. We looked at 2-socket, 4-socket, 8 sockets. In terms of software, we look at many parameters in over 30 different tools and different implementations. We conducted thread scaling studies, same with the shards and memory allocation. So realize that what we did is, we started running something that actually took us 30 hours of the beginning, and we, very patiently and incrementally increased, fix all the variables and worked on one at a time, increasing, say, for example, from 1 thread to 2 threads, all the way up to the max for that architecture. So it was a very painstaking process. And I hope from this description you can appreciate the fact that it's something that requires hundreds of different runs to test the effect of the permutations of architectures and setups, because it's really a multidimensional problem. So much that this sort of monumental benchmarking effort is something that is seldom or never undertaking at data centers, because data centers often don't have all these different options available. And the HPC teams are supporting their researchers, and the researchers are doing the science. So this is an area we identified where we thought we could help. And it was quite a painstaking and long process, but we're glad we did it, because at the end, and in fact, in a matter of the last 3 months since we've been in this quarantine, we were able to arrive at 2 new architectures that improve performance a whole lot more. We were able to find an architecture that actually processes this in under an hour. Now, we're actually breaking records here. We're delivering performance with a CPU-based architecture at the GPU level. So if you think of genomics performance levels as a spectrum, all the way from minutes to 150 hours, our GOAST solutions are at the very end of the very optimized end of that spectrum. And see, the beauty of having done this in such a comprehensive manner is that we understand all the building blocks that go into. We can, dial things up and down so that we can offer architectures that can be customized. So if your priority is top of the level performance, we have an architecture that does that. If your priority is affordability, we can do that as well. And our most affordable architectures are in fact still some of the highest performings out there. Okay. All right, so that's a feature of GOAST. GOAST also supports high throughput volumes. We can support all the way from the single research lab with a single sequencer to the population level efforts processing hundreds of thousands of genomes per year. I'm going to show you a table here, and each row represents, two of those architectures, the GOAST architectures. And on this column, I have the number of genomes per node per day you can expect for that architecture. Now notice that both of the architectures can support, volumes as high as 100,000. What changes is the number of nodes that you would need for that. So achieving high throughput, we're doing it in two ways. One, we know the nodes, we can figure out how many nodes you need to support that volume that you need. But also we have figured out ways to maximize hardware resources so that you can process many concurrent samples at a time. So we can do high throughput runs at the same time. Also, we're, as I've shown you, we're accelerating omics analytics. As I've shown you, we've done, tremendous progress in terms of being able to increase the performance of genomics. We're also very excited about that, some of the preliminary work we're starting is showing that this same architecture is also improving transcriptomics and other bioinformatics sequential pipelines. We're also increasing usability in that we're providing pre-configured pipelines that leverage all these, the discoveries from our systematic testing, and also are offering pre-tuned hardware with the optimizations to be able to deliver this sort of performance. Now, we are securing data privacy in that by running GOAST, an improved, an optimized setup, you can run on-premise and on hybrid environments, which circumvents many of the security issues plaguing the public cloud. All right, so now let me end by showing you how Lenovo HPC is enabling COVID-19 research around the world, many of the data centers around the world. Let me show you two examples. The Beijing Genomics Institute is one of the largest genomics institutes in the world. They're spearheading the process. In fact, they were one of the first to create a testing kit for COVID-19 infection, for SARS-CoV-2 infection. And so at the beginning of this pandemic, we partnered with Intel to donate a large cluster of GOAST 1.0 servers powered by Intel Xeon processors and fast storage. They can process about 95,000 whole genomes per year with that cluster. And these are some of the research they're spearheading in COVID-19 and the COVID-19 topic. They're looking at the virology of the virus. They are identifying determinants of susceptibility. They're working on detection kits and improving them, studying the origin of the viruses. They're involved in vaccine design, studying the immune response, drug targets, and also elucidating the molecular structure of the virus. Also, another one of our customers is also using our solutions. The University of Birmingham is one of the UK's leading research universities. Their supercomputer is powered entirely by Lenovo. They're using our water cooling technology as well as Lenovo boxes powered by Intel Xeon processors. And they are part of this consortium, the COVID-19 Genomics Consortium in the UK. And as such, they are developing portable sequencing solutions. They're tracking infection spread, looking at the effectiveness of treatments, and identifying potential vaccine targets. So as you can see, Lenovo is leveraging the GOAST Genomics optimized servers to help data centers around the world. We have really focused on delivering increased throughput, accelerating the workflows because we know that's where the bottlenecks are, increasing usability, helping you keep your data safe, planning the HPC resources more effectively. And our hope is that with this contribution and by doing so, we're empowering the scientists in the front lines of the COVID-19 research to accelerate their path to discovery. If you want to learn more, I'm going to leave you with some links you can follow. And of course, feel free to reach out if you need to learn more. Thank you.